This is a conversation with George Church. He's a pioneer of human genome sequencing, the architect of CRISPR and multiplex editing, founder of nearly 50 biotech startups, and the mind behind projects as audacious as virus-proof cells, de-extincting mammoths, and reversing aging. We talk about the limits of scaling SynBio, why AI is the next engine of discovery, the real economics of gene therapy, and the lessons from Ginkgo and 23andMe. This is the Acc Podcast. Please enjoy. Thank you so much for joining me. Your lab is obviously known for spinning out many biotech ventures and has been called the Bell Labs of biology. In 2018, you spun out 16 companies alone. My first question is about your lab, what you're excited about, what people are working on, and some of the technologies that help you get up in the morning.
Yeah, it really is a remarkable time. I've read enough about Bell Labs to you get the feeling, and I'm sure there's differences, but it's partly just the time. It's the time, the era that we're in. It's exponential technologies, not just in electronics, but in molecular biology, and then the derivatives of molecular biology, which are, developmental biology, which actually makes it into clinics. So to give some examples, reading and writing DNA has come down in price and up in quality about 20 million fold in just a short period of time. We can now do not just editing of genomes, but multiplex editing, where we make lots of changes. And examples of uses of multiplex. Some people might say, well, why would you want to change more than one gene at a time? And examples are, we've made organisms resistant to all viruses, even ones we've never seen before, by changing the genetic code, which required thousands of edits. We've made pigs that have 69 edits in order to make them compatible with use in transplantation into human patients. And we've explored mostly liver and kidney. We now have our longest patient, I think, is January 25th of this year to May 16th now. And so it's getting close to the point where we are already in the process of scaling up and doing FDA-approved clinical trials. And then the third example where you can use multiplex editing is for increasing the diversity of a species, either by bringing things from distant parts of the world or distant in time, as far back as 2 million years. You can restore diversity that's been lost and maybe even make things more diverse than they ever were. We're trying to engineer resistance to various pathogens into wild species like ferrets and elephants. Those are some of the things that are pretty exciting. All these things intersect with artificial intelligence and the electronics revolution. We're even making molecular transistors and electronic circuits that, large-scale integrated circuits with thousands of molecules in them. Published a PNS paper on that. So they merged literally at the material level, but also at the intellectual level. Almost all my companies and my lab use AI, machine learning, to design new proteins, new viral capsids for precise delivery, and so forth.
There's a lot going on.
Which may come out in the rest of our conversation.
I'm sure it will. I totally agree with you. I think the scale and the precision of multiplex editing that you guys have made possible and that you're pioneering in pigs is really the tipping point for engineering biology. It's almost like it has that same modularity as software, which is insane to think about. We had the .com revolution. We had this entire revolution with tech. And we're seeing Moore's law being applied to biology, which is crazy and so exciting to think about. You touched on the cost of genome sequencing going down, and that's something obviously you pioneered. That's something I'm very excited about, is the cost of these technologies going down. How can we begin to think about scaling biotech ventures and more specifically scaling SynBio adjacent companies, since I know that is a challenge right now. And that is leading up to my next question. There's this growing view in many of the investor and maybe founder community that SynBio hasn't really delivered its AWS moment. It's like a generalized platform that unlocks predictable, scalable outputs across many applications. What are your thoughts just generally about the scalability of SynBio as a whole? I know a lot of investors have expressed some skepticism around scalability bottlenecks, like the biology maybe is a little too stochastic or unpredictable. manufacturing constraints. I know it can be very expensive, especially when working with cells and tissue cultures to scale at that level. Really curious about your thoughts. The reason I'm asking is there's a really robust SynBio community here at Harvard. Many kids are excited about the field. And I think when you think about SynBio, you almost have a tendency, or you're almost forced to think about how that intersects not just with research, but with producing products and starting companies. My question to you is, what are your thoughts about the future of SynBio as it's integrated with more industrial output?
Great question. Let me unpack it a little bit. One of the sub-questions was stochasticity, reproducibility, and scalability. Stochasticity, or the apparent irreproducibility, some of it had to do with the fact people weren't very careful about their strains. They would pull strains that came from different passages. One source of stochasticity is mutation and epigenetic change. I think we've gotten better at controlling that. We've got better at reducing. We finally have our hands on all the knobs and switches. And so if we want to have something retain its exact DNA sequence and its exact epigenetic state, it's getting to be within range for us. That's one source of irreproducibility. The other is that they're single molecules, so they should be stochastic. I mean, that's part of quantum mechanics, or even just distribution of matter. But it turns out that circuits are starting to get stochastic too. When you put a dopant atom, like boron, into silicon, the number of dopant should be at low concentration. If you shrink the volume, that atom gets down to like an average of 1 or 2 atoms per chip element. That means if the average is 1, 37% will have 0, and 28% will have more than 1. That's stochastics. And you either have to make your circuit or your system tolerant of those, or you have to eliminate them. One way to eliminate them is proofreading. John Hopfield, who got the Nobel Prize for his contribution to neural nets and AI, could have also gotten one for his kinetic proofreading theory. Almost everything in biology involving polymers has proofreading. You can do something stochastic and then proofread out all the errors. So that's addressing some of the stochasticity. Scaling up, biology scales up quite nicely. It's very fault tolerant, as long as we're tolerant of the faults that it's tolerant of. For example, you can do amazing things in biology where you don't have to micromanage it. So you can take the optic cup on login from an amphibian tadpole, move it into the tail where there isn't supposed to be an eye, and it will spontaneously wire up to the nervous system. It will respond to external visual cues and connect to muscles to do avoidance reaction. All of this, you didn't have to micromanage. You didn't have to write a line of code for those actions, including all the developmental biology. So even though it's an incredibly complicated system, I would argue that even a tadpole brain is more complicated than anything a human has engineered. A human brain is way out of our league. There's this incredible complexity you need to accomplish things. There's atomic precision. So a huge fraction of biology is absolutely atomically precise. You take a look at 2 copies of the same enzyme and proofreading. So I think it's a beautiful time now to get, I think one of your last parts of the question were, should people get excited about this? Definitely. I mean, this is something where you can make things way more complicated than your biggest GPU cluster. We're proud of our GPU clusters, but you can make, something even better than electronics because it's fault tolerance, error correcting. It's self-repairing, sometimes for centuries. We've got bowhead whales out there that are centuries old, and it's self-assembling. You don't have to say, I need a government grant and I need hundreds of people to build a forest. The forest is already there and it's building itself. You can even rip it down and it'll rebuild itself. It's nice when people plant trees, but the fact is for millennia, they've been planting themselves. The cost of any atomically precise material should be on the order of dollars a kilogram, no matter what it is, whether it's something as smart as the human brain or as dumb as a piece of lumber. So anyway, it's a great time because we got this exponential change. Every year, the intellectual and financial landscape flattens, which means it's open for new entrants who are just as expert as the experts were for the last revolution.
Yeah, I think the stochasticity part, to maybe speak to that, I think it's a really compelling analogy that you've given us, that if we accept stochasticity as a fundamental property across both biological and even silicon systems, it reframes this challenge of not how do we eliminate noise, but how do we engineer systems that are almost noise aware or resilient.
Exactly. No, it's
And
true. And in addition to the stochasticity of manufacturing, there's stochastic post-manufacturing. So, for example, as you get to nanoelectronics, they become very radiation sensitive. And biology's already got all kinds of repair systems that are really only limited, as far as we know, by the fact that our ancestors were cheap. That is to say, they had a limited amount of food, and why would they spend extra food and ATP on on repairing something when they were going to die tomorrow anyway? So we're still amazingly good at repair, especially the long-lived species like humans and bowhead whales.
Yeah, and I think that's a really poignant question for the aging folks. They're trying to repair function in humans, and a very similar belief they have that didn't evolve to die, that these are just gaps in our genetic code. They're gaps in our biology that we should try and supplement to the best of our ability. And it's interesting that you come at that from an evolutionary perspective, that we didn't evolve to do that because there's, at that stage, no point of doing that. We only live till 30, 40 years of age.
Well, for rodents, we only live for 1 year, if we're lucky.
Yeah, exactly. That's a super interesting thought. I want to touch back on our discussion on scalability of synthetic biology, and I totally hear you. But as a student reading the news, going on Twitter, I'm hearing a lot about some of the companies in synbios, and more specifically, some of the public companies. And I think Ginkgo is a good example of that. There was a lot of excitement at one point about Ginkgo, and it was once considered the AWS of cell programming. But from what I've reading, it seems developments have raised questions of whether synbio can be successful at that scale. In 2024, they reported about a 10% decline in annual revenue. There were some net losses, like 550 million, and some layoffs as well. And I've been noticing the stock price at a low right now. I don't know if you've been following them. I'm not sure how much you know about the company in specific. Given some of those challenges, maybe you can comment to Ginkgo specifically, but I think I'm more curious about what insights can we maybe draw from the current limitations in some of these companies. Do you think these setbacks are indicative of broader systemic issues with the field, or maybe just company-specific or strategic missteps?
Well, I do know a little bit about Ginkgo. I knew all the founders when they were graduate students. Tom Knight, he was one of the people that was very welcoming. Furthermore, they acquired, thankfully, some of my companies, including Gen9, which was, I think, one of the premier DNA synthesis companies, and it's still I think it's one of the jewels in their crown at Ginkgo. Now, when any company loses $500 million, it could indicate great health. That is to say, they have good management that knows how to downsize. It may mean they have billions of dollars they haven't gotten rid of yet. So I actually I think it's could be quite healthy. They did pick something that was practical and pragmatic at the time, making sort of biochemicals, sort of, flavors, fragrances, other slightly high value chemicals. So not the most commoditized things like, say, biofuels, but a little bit higher value, but not as high value as, say, erythropoietin. When we think about the entire biotech field, we have to include all those different valuations, some of which have very high profit margins. Some of them weren't classified as synthetic biology because they were part of the biotech revolution, which I also participated in. I was one of the first employees at Biogen, which with Amgen and Genentech kind of established that. In a certain sense, that was part of synthetic biology. It was the very first inkling that we were going to be able to do that. But now it's definitely a whole nother entrant in it. So we have the biotech, molecular biology, the biochemistry metabolism of Ginkgo. I think they're doing flexible manufacturing where they can reconfigure. I think that's all terrific stuff. Then there's developmental biology like we did at eGenesis with pig organs. There's single editing and multiplex editing. There was just in the news a case where a very desperately ill infant they made a CRISPR for it. This was a one-off with NIH's approval and FDA's approval. I think it's a whole new thing. If you can make one therapy for one person, that might actually reduce costs, of one way of reducing costs. But I think the next thing after all of these things is the merger with AI. And essentially almost all of my last 7 companies have included AI deep in their business. I think that will enable almost all aspects of biology and medicine. The most extreme is Lila.ai, which I'm chief scientist. just came out of stealth mode after a couple of years, with hundreds of millions of dollars already in tow. I think their lesson from the AI field is learned over and over again. If you can generalize, you're going to do better. They're generalizing it to all of science, or at least to all of science that can fit into a warehouse-sized room. It's amazing how they synergize, how things you learn from, the chemistry of catalysts is applicable to the kind of chemistry of biological systems, how biochemistry translates to transcription factors, which translates to developmental biology and so forth. There's a lot of synergy, and you might as well use that. You can build a huge proprietary database, which is bigger than all the existing databases that most companies get by scraping the internet. That's how most AI companies do it. This poorly annotated large data set. So you go to PubMed and it takes an expert really to, even when we have good natural language processing. So I think that's the next revolution. Ginkgo has been foundational in building in that direction, and hopefully it will participate as well. It'd be great.
Totally agree with you on the AI revolution in bio. The drug development pipeline is already very long and obviously very tedious. And so I think there's huge potential in the early stages of drug development that can really speed up that process. And I think companies are already beginning to implement. Question for you about companies that focus solely on, AI drug development or generating lead candidates at the very early stage. I'm curious about how successful you think these companies will be if they want to eventually get acquired. I have this suspicion that these big biotech companies, they're already creating their own in-house models for optimizing these processes. If a small company wants to come in, do you think these companies will outsource this? And what do you think that means for the model for these companies?
Well, I mean, there's a lot of different answers to that. I think outsourcing is very common. The bigger the company, sometimes the harder it is to innovate. Their source of innovation is being clever about who they acquire and then making sure that they nurture them. Because if an acquisition is too easy, if it's easy to throw $300 million at something and then put it on the wall, or it doesn't do anything, then that discourages the ecosystem a little bit. But being acquired is just one way you can succeed. Some will keep doing it on their own, and as soon as they start getting revenue, then they no longer dependent upon investors so much. Some will become vertically integrated, where they de-risk by making sure they have their own supply chain in-house. There's just a lot of ways to succeed, and there's a lot of ways to fail as well.
Yeah, that makes sense. I can imagine larger companies are often getting bogged down by risk aversion and all their internal processes. Using strategic acquisitions as a form of creative innovation is a really interesting workaround, I think. I wanted to now shift towards genetics. I think we focused a lot on AI and the synthetic biology field as a whole, but you are quite famous for genetics. You played a central role in driving the cost of genome sequencing from $3 billion from the first human genome project to under 600, which is incredible. My question to you is, why do you think genome sequencing scaled so successfully? And yet, follow-up question, what are your thoughts on the current 23andMe bankruptcy? Do you think that's a consequence of the technology they've created, or data privacy, or certain partnerships that were not strategic? Just your general thoughts.
Okay, I'm going to divide that into two questions. First, what caused that incredible revolution in kind of where you got a Moore's Law-like effect, but in a shorter period of time? So Moore's Law might have taken 40 years to cover what the sequencing field did in about 16 in terms of doubling times. And I think it's more than you said, because it was 3 billion for one genome, but typically for clinical use, you need two genomes, your mother's and your father, or your mother and father contribution to your cells. And doing two is more than twice as hard because you have to keep them separate. You don't want it to like cross over from mom and dad, because then that actually changes the interpretation. So let's say it was 6 billion plus, if we had done it, which we didn't. Today, I think the cost is probably $100. The lowest street price is probably in the $250 range, depending on how you count subscriptions and things like that. So it's gone from basically 6 billion down to a couple hundred. That what happened, I think, was partly the Genome Project baked into it some conservatism, which I think did not serve it well. I objected from the very first meeting in 1984, the Department of Energy meeting. And our message was if they wanted to estimate mutation rate, they needed to know at least one genome as background information. And they loved that and immediately started funding it, mostly internally. I got one of the first DOE grants in 1987. NIH didn't kick in until 3 years later. They said, well, we can't let Department of Energy, of all places, do maybe one of the coolest things in biomedical history. They weren't sure, but they said, let's not take any chances. What was baked in was this conservative attitude of let's use whatever technology is available in 1990 and just scale it up. And maybe if we scale it up, we'll learn something. And maybe if we automate it, we'll bring down the cost. But the problem is the automation has a tendency of just replacing a person with a machine, and all the reagents and the consumables and machine amortization is more or less equivalent to the human version. It doesn't really save much money. They didn't fund technology development for most of the genome project from, let's say, 1990 to 2004. I think the DOE was on a pathway of innovation. They had a lot of robotics for the descendants of the Manhattan Project. They had the first GenBank, or they had single molecule fluorescent sequencing started. They had chromosome sorting. They were really into technology. But as soon as it hit NIH, then it became, where's it going to be done? Who's going to do it? How are we going to split the $3 billion? There was no talk about, well, let's get it down to less than $3 billion. It was just who gets the biggest slice of the pie. And I felt that we should bring the cost down to at least 1,000, if not less. That didn't happen until the project was over, and they realized we got to sequence more than one human genome here. They've got to be diploid, they got to be high quality. We basically threw out all the technology that was developed through the Genome Project. Nothing remained that wasn't already there at the beginning. Shotgun sequencing was there thanks to Fred Sanger, but even dideoxy disappeared because what happened was a complete reconfiguration. One was nanopore sequencing, which was single molecule, and the other was multi-molecule fluorescent sequencing. And they were just radically different. The single molecule, none of them involved electrophoresis, and the imaging, the single molecule kind of involved the shrinking, which brought down the price of electronics so much. But since electronics had already done their shrinking thing, we could benefit from just going straight to the frontal line where electronics had already paved our path for us. And so we shrank faster and benefited from all the microfabrication and so forth. I think that explains most of it. Anyway, let's do the 23andMe. I was involved in that since before they started. Ann Wojcicki, Linda Avey, and Esther Dyson were the board. I think it was a brilliant start. They pivot. They started, I think, with the idea of using Illumina chips for genotyping, which was a lot cheaper than DNA sequencing. At the same time that I was working with them on this genotyping was like, 1 million very common polymorphisms, which weren't likely to be associated with disease. I was also working with GNOME to do whole genome sequencing. And of course, the difference was the 23andMe started at about $1,000, but wasn't about disease. And GNOME started at, I think, $350,000 and gave whole genome sequences. I think it was inevitable that they would be outflanked by genome sequencing. At least I felt it was inevitable that the price of sequencing would come down to about the price of the chips for these common polymorphisms. And the other thing was they had to keep pivoting their business model. So it started out, it was kind of like do it yourself, get to know genetics. And then it pivoted, included more Ancestry, but it was competing with Ancestry.com. Pivoted to, let's ask some interesting questions and build up a database of phenotypes, but they weren't taking medical records. That pivoted to maybe we can help the pharmaceutical industry. And then finally to maybe we can be a pharmaceutical company. I think all those pivots didn't give time to become expert at any one of them. And I'm not even sure which one they should have stopped on because, I've always voted, my main vote has been get the carrier screening working because that'll save the world trillions of dollars and lots of pain and suffering. But it requires looking for rare alleles. They were looking at common alleles, which in principle are in linkage disequilibrium. But the problem is, is that those rare alleles can be next to any common allele. So they're present in essentially every haplotype. couldn't really deliver the kind of carrier screening you need to help everybody get healthy babies. Nevertheless, I think there is still movement, like the Human Genome Project #2, which I'm involved in, to get the whole world sequenced at high quality and immediately get them the privacy, but the results they need to have healthy family members. I think that day is coming, and it's already at the point where nobody should have to pay anything. Every genome that gets sequenced results in, on average, 10 times return on investment. It's not like starting up cell phone circuitry. It's already there.
No, that was excellent. Really agree with the carrier screening thing. I think that has a really clear clinical utility, a very clear market, arguably less regulatory hurdles than some of the D2C health prediction efforts they've pursued. And I think it aligns a lot better with physician adoption, like OBGYNs, fertility clinics. It's a huge market, and even primary care could have been really natural partners there.
Yeah.
Do you think maybe if 23andMe had doubled down on medically actionable carrier testing early on, they could have transitioned from a novel product to an actual infrastructure layer in preventative genetics, which could be a huge revolution in how we use information from our genomes?
I would have liked to have seen, in fact, but I'm not going to say that it definitely would have worked because, well, first of all, they would have had to not only change business model, but change their technology platform, which they didn't change during the whole thing, to sequencing. And there was competition in sequencing. And it was, and the price wasn't coming down that fast. I think at this point, they're about the same. The other thing is, nobody else has really cracked the carrier screening problem yet. Okay, you should be able to get a tenfold return on investment. That's my techno-economic analysis, amateurish, and nobody's managed to get it to work. But I like the way you jumped. I mean, you just immediately jumped from what I said to exactly the right place, which is it is lower regulatory problem. It's not like I don't want to avoid regulatory because I love the FDA. I have very good relationships with them for our pig organs and our gene therapies and so forth. They love innovation. It really isn't hazardous. Let's say you have a false positive rate and you're waiting until they're fully adult and they present in the clinic with some problem and you say, oops, you got this genetic predisposition. If it's not totally clear, there's a lot of modifier genes. It's not always simple, even if they're so-called monogenic Mendelian disorders. There's these modifiers and environmental things that you could do, let's say, bilateral mastectomy and ovarectomy in an adult that hasn't yet had children. You've basically said they're not going to be having children the normal way, at least, and they're having these risky surgeries. And then a year goes by and the front page of the New York Times says breakthrough in breast cancer genes, and you didn't need those surgeries after all. So that's a tragedy for that kind of false positive. But if you do a false positive at, say, early stage premarital matchmaking, there's no harm, no foul. You could have false positive, 20%. I mean, a ridiculously high false positive, and it just means that you still got 80% of the population to date or to marry. The challenge is getting that matchmaking thing lined up with heavy-duty genetics. When you're matchmaking, you don't necessarily want to think about genetics, although it is one of the main things you should think about. There's good worked examples that we should pay more attention to and celebrate and study, which is Yesherim. Yesherim has been around since, I think, mid-'80s, and it has very much reduced, maybe tenfold reduced certain diseases, maybe their top 9 diseases that are enriched in the Ashkenazi Jewish community. And they've got very high acceptance and millions of people, but not billions. And for some reason, it hasn't generalized to other. So it's probably among the most successful attempts at carrier screening. But anyway, we need to study this. It's a very interesting sociological, technological, ethical case study.
Yeah, I really like that case example. Correct me if I'm wrong. Just for viewers who might not be familiar, they anonymized carrier status and only revealed compatibility. And by doing that, they reduce the incidence of Tay-Sachs and other disorders by an order of magnitude without stigmatizing the individuals. And because it's a community-wide model, it's standardized and deeply integrated into culture norms, it worked. Yes. Is that clear?
I think that's a very, very clear statement of the situation. And I think you could make it even more anonymous than it is, because right now you typically do pairs. This is not the only way that they practice it, typically pairs. And so so you actually might have already fallen in love, and then you have to make the tough decision whether you're going to marry anyway and just do IVF, be very careful in other ways. That's unfortunate. And it also does, and if you break apart, then that stigmatizes both individuals. It means there's something wrong with the two of them, depending on how well educated the whole community is, which is fortunately pretty well educated. But if you did it by matchmaking at an earlier stage, you could have homomorphic encryption query, which is a way that you can ask whether this person is compatible with any of them, and then you just match them up. And you don't say whether this person is a carrier or whether any of the other people are on the carrier list. You just match them up. And you're going to randomly leave a bunch of them out. So that doesn't mean that they have it. It just means you can't match everybody at once. And I think that would be even better. But I think what they have is adequate. It's just the question is, how do you adjust it to other cultures, or how do you adjust those cultures to the new norm? And it's just a fascinating and very important medical, it's one of the best medicines we have. And it's also one of the least risky and cheapest. Some of this is subsidized, but it's in the $100 range. And compare that with $4 million, which is the cost of a gene therapy. I was appalled at the direction that the price was going. It's gone up eightfold in 8 years. And so now 4 million is the new norm and I feel that the better use of our resources is to keep gene therapy for really common diseases like pandemics and aging, where you can get the price down to sub $100, whole genome sequencing for all the rare diseases. Then boom, you've got both of them, and you still might need a little bit for people that are already born or people that escape the opportunities for diagnosis.
Yeah, wait, that's really interesting. So, why do you think that these specific applications would be a lot cheaper? My understanding of the gene therapy, one of the reasons that the cost is so high is because a lot of them are targeted towards rare diseases or congenital disorders and smaller population. You need to recoup your investment. So, is that the logic that you're trying to get at, or not
Once
really?
again, you absolutely brilliant and jumped to what I think is the answer, which I didn't jump to nearly as fast as you did. It's probably economy of scale. You've got fixed R&D costs and clinical trial costs. The denominator is very small, so you're going to have $4 million a dose. I mentioned there's this new case where one person completely individualized therapy. That probably reduced the R&D costs of going through a full clinical trial, but it probably had slightly higher costs of doing that custom design. But that could be brought down. I mean, I can imagine that coming down. And so that might be a good thing. But the reason I say it's more than just a theory that it's economies of scale, we actually did the scaling during COVID-19, where 6 billion people got what was clearly gene therapies, single genes encapsulated in the normal gene therapy way, intramuscular injection. And so that showed that it was safe and effective, and in a certain sense, de-risk all subsequent gene therapies. But there are very few things. I mean, that pandemic was once in a century, hopefully. But the thing that will persist, and maybe even bigger, is age-related diseases. That's going to kill 90% of us, and it impacts almost every form of medical wellness. So I think that is the next thing that we should be aiming gene therapies at, and other things that would be classified as orphan drugs. We can nudge those over into this really common disease category, and then handle almost all the rare ones with genetic counseling.
That is a really interesting thought. I'm going to be thinking about that for a while. No one talks about that
Right.
pandemic. We think of it as such a risky, unstable technology, but it's really not. Like it has been de-risked so many times before, and I think now people are beginning to get really excited about gene therapy. As of now, we only really have it for monogenic disorders and for somatic mutations. There people are talking about using CRISPR and gene editing for germline editing. I don't know what projections you personally might have for that and what your thoughts are, but it might be one of the last questions that we get to before we ended the conversation.
Right. It's a good conversation ender. There's general global consensus, at least among the experts. The experts love to declare consensus when they really haven't consulted that many people. So I'm not declaring consensus, but that we shouldn't do germline in humans, at least not right now. We do get germline editing into human beings in the sense that we do germline editing of pigs, 69 edits to get their organs ready. Then that kidney goes in and hopefully it will last for the rest of their lifetime. That shows some of the advantages of germline, which is you can really do great delivery. You can deliver to every cell in the body using normal developmental biology. It's not particularly expensive, scales well. In fact, we can scale just by breeding pigs the way we breed pigs for food. So that's the advantage. The downside, there's really not been articulate. I have defended it at least as something to be considered rather than rushed into. I've defended it more than most people do, but there's not a good use case. Most things that are fluctuating the population means that you can solve it either with genetic counseling or gene therapies. So you don't really need germline. The place you need germline, and places where you need germline, is when it happens really early in development. Let's say you're microcephalic, you have a tiny brain when you're born. It's too late to fix that with a simple drug or even a complicated drug. But that could have also been fixed by preconception or pre-implantation analyses. For germline, if we get good enough at animal germline and we're using the animals for medicine, that might change the norm expectation. But there's got to be some application where it really makes a difference. The double whammy is enhancement. It's like enhancement is okay if it's electronic enhancement of the human, that's fine. Locomotion, that's fine. Even vaccines and caffeine, those are enhanced, those are fine. But then you combine it with germline, and that's the one thing that might actually make germline useful if we had some enhancements like the ones I just listed. There are people walking around that have PCSK9 and CCR5 that supposedly have some advantages for cardiovascular and infectious disease, respectively, and they could probably be engineered to be even better. But you might be able to do those somatically. A lot of times when you have this ethical issue, there's really some practical engineering issue as well that people just can't articulate. The practical engineering issue here is that 8 billion of us have already missed the deadline for germline engineering. We want our hard-earned money to go into making us youthful for, a century. We want to be like 20-year-olds for the next 100 years. We're not going back to the embryo stage to do that. The other practical consideration is that if you want to test a drug, you need to go through the full lifetime of that drug. It's not always done, but it should be. For example, if you want to do aging, well, then that might be a 30-year time. That's hard. If you want to do aging starting from germline, it's now 80 years, 100-year clinical trial, and you might want to see whether it affects the next generation, which makes it like 200-year, and nobody can afford that, and it's just impractical. So that's another aspect of it. While if you do a reversal of an age-related disease like cardiovascular or neurodegeneration, that could happen in weeks. That could be an absolutely normal clinical trial. And in fact, our clinical trials are getting shorter and shorter. So COVID was 11 months, and this customized, individualized was even shorter. So we may be moving to short clinical trials. And so these really long clinical trials, which you would need for aging or embryo engineering, are not going to be competitive. So those would be my two ways of looking at why we're not doing germline, and probably why we should double down on getting really good at delivering to the whole body, which is one of the things that germline does very well. And there's absolutely no reason why we can't do that with somatic adult therapies.
Yeah, great points, especially about the length of the clinical trials. I haven't even thought about that. But yeah, that makes total sense. You would have to go through an entire generation and then make sure that those edits didn't affect the subsequent generations. And so that would be practically unfeasible. I don't think people are talking about when germline editing is not ethical. We shouldn't be changing our embryos. We don't know how it'll affect the child. How will that even be regulated? How will it become approved? What evidence does the FDA need to see in order for that to get past those regulatory hurdles? And You mentioned 8 billion of us are already past that threshold where we can't see the benefits of germline editing. I think that further supports why aging and longevity research is getting so much hype right now. If I'm 20, when I start doing research in longevity, maybe I'll be able to expand my own lifespan. And that is a very fulfilling prophecy. It's very exciting. I think that's why a lot of young people are excited about that field, because of the possibilities of tangibly seeing that impact on your own lives rather than looking back in time. I
Yeah.
know we're just about at time here, so I really want to thank you for this conversation, Professor Church.